So we're going to talk about several things. So first, we'll go through the virus basics and we'll refresh the knowledge of what we know about virus. And then we will introduce some common methods for detecting virus. And then we will discuss two methods. The molecular methods that enable detection of specific viruses by the means of detecting their protein or by the means of detecting their genetic materials. And then we'll move on to discuss some of the recent advances in those molecular detection methods. So speaking of virus, so viruses in general are made of a protein coat, which forms the outside the shell. And inside the cell, it has its genetic material kind of protected in this shell. This coating protein, other than serving as a protection to the genetic material, also serve the function for docking onto its host and for entering into the cell. And inside the shell, in some viruses, it also carries some other enzymatic proteins, such as integrase, as well as some proteases or transcriptases. And those are for the amplification of their genetic materials and for their proliferation. However, the virus never have a complete set of all the tools necessary for its proliferation. So it cannot proliferate on its own. It always have to get into a host, such as a cell, and to utilize the cellular machinery in the environment to proliferate. And so given these features, so there are several ways to detect the virus. And for example, physical measurements, we can focus on the size of the shape or morphology, and we can directly visualize it to detect it. Or we can focus on the cytopathic effect, which are caused by some of the enzymes inside the host cell. Alternatively, we can do the molecular detection of its proteins or its genetic materials. So to visualize the virus, it's not very easy. And because it's extremely small, it's about an order of magnitude smaller than the bacteria. And some are a little bit larger and some are even smaller. I mean, in general, it's just a bunch of proteins. So it's on the order of proteins right here. So with conventional light microscope, we cannot see virus. We need microscopes such as electron microscope to be able to see them, which provide a much higher magnification. And there are two modes of electron microscopes that are commonly used to visualize viruses. First one called scanning electron microscope. So it works in a way that we normally see things. So we have light shining on it, reflected back to our eyes. And so here, in a very similar principle, we have electron forms the beam and shine onto the sample, and the reflected electrons get collected by these electron detectors. And so to image the features of these biological samples. Another mode is called a transmission electron microscope. It's in a way that we see things by seeing the light and passing through it, and so then they leave the shape of the things we want to see. And so here electrons go through the biological sample and get detected by the screen at the other side of the sample. Typically, a biological sample neither reflect or block the electrons. And so we have to fix the biological sample in a particular way and to stain it with heavy metal, which then gives the contraction we need for electron microscope imaging. In fact, many of the pathogens were first identified under electron microscope. And so here we have E. coli, we have influenza viruses like Ebola virus, and here are the coronavirus. And some of these pathogens even got their name based on their looking under electron microscope. And so the coronavirus, for example, got its name because it looks like little crowns on the surface, when people first see them. So that's electron microscope-based visualization. So another thing that I mentioned is called a cytopathic effect. So this is based on the fact that so when the virus starts to proliferate inside the cell, it drains the cellular resources, and that's kind of how it kills the cell. And that will trigger the cell stress response. And so the cell will start to look unhealthy and look different. And this phenomenon is called cytopathic effect. So the way it works is that we'll flush the lung where we know that the infection occurs. And so the liquid will carry off some of the extracellular content, including the virus that we're interested. And we have this liquid, then we pass on this liquid into the cell in vitro in the dish, and we observe the morphology changes, if there's any, to the cell. So here are some examples. So in normal cell culture, the cell will look flat and healthy and grow very happily. And so when the cell gets infected, it will start kind of get a little bit unhealthy and starts dying. And as it grows, it will start to show gradually more severe signs of stress and dying. And that's how we know that there's something causing the problem, and that's probably the virus. And in fact, this is the method that they first used to confirm that something is happening, that we have the virus from those pneumonia patients back in January. Those two methods are cool, but as you can see, they're not very specific because many viruses look very similar under a microscope. And many viruses can cause the same symptoms in a cell culture. And critically, different strains, the strains and bears the different resistance to drugs. And they can bear the similar stress on the cell and make the cell show similar phenomenon. So these two methods are not going to tell us which is a resistant strain, which is not. It won't tell us certainly a new virus strain either. And so we need a different method. And so the first one we're going to talk about, and based on the molecule detection, is through the detection of the viral proteins.